Good morning, everyone. Welcome to the fourth day of this biotechnology capacity building cell crash workshop organized by Ramakrishna Institute in association with and sponsored by Gujarat State Biotechnology Mission Gandhinagar. In first session of today's workshop, along with us, Dr. Nehal Shah. Dr. Nehal did her BSc, MSc, MPhil, PhD in the subject of zoology from Vietnam South Gujarat University. She possesses a 5 years of teaching experience at UG as well as PG level. She has published 4 research papers in a journal of national as well as international repute. She has attended more than 15 conferences, again at the state, national as well as international level. She possesses her own website which is related to female problems, and she did her PhD on a subject which is known as the premenstrual syndrome. She has delivered a couple of lectures at Biceps Sandhan Center. Madam, on behalf of Ramakrishna Institute and GSBTM, we are welcoming you over here.
Good morning, everybody. Am I audible? Can you hear my voice? Okay. A very warm welcome to you on the 4th day, and I'm extremely thankful to GSBTM as well as the organizers for giving me an opportunity to be with you today here. You have been studying a lot of topics all these 4 days, and what you have missed till the date was zoology perhaps. That was not covered, so I have tried to look back into little portions of zoology which might be useful to you for your exams, particularly a topic which I always get a complaint about. It's very boring and it's very lengthy, we cannot remember, and that is taxonomy. So I would like to give you a few tips on taxonomy, as well as we'll do little clarification on the concepts related to taxonomic studies. It's very essential to understand taxonomy, how, what, when, that we shall just move on, you'll come to know. But you can see on the board that there is a slide which tells you that luck happens when preparation meets opportunity. So you need to do a lot of preparation to fulfill the opportunity, then only you can just crack your exams in the best possible way. And for that reason, we need to remain concentrated for the next at least 2 hours, right? So our topic for the day is going to be animal taxonomy, very basics, fundamentals. I would also like to share this kind of study material. Soon it will be given to you. Ali sir, please. This particular 3 pages will help you, revise and give you all the basic tips related to the MCQs which you might come across, particularly invertebrates. I've gone through your syllabus as well as your the MCQs that you're getting, and in that mostly the concentration is more on invertebrate side rather than vertebrates. So my today's focus will be more on the concepts related to taxonomy, how you can deal with this particular topic in a very short period of time when you are preparing so many subjects at a time. So this particular pages will help you revise the whole taxonomy in the shortest period of time. Not wasting time further, before we start with animal taxonomy, a very basic question, rather a very, I would say it sounds stupid to ask what is animal at this stage when you are completing your graduation and sitting in front. Fundamentally, plants are the ones which prepare their own food by photosynthesis. Animals cannot do that, most of them. We don't take the exceptional cases presently, but most of them do not prepare their food on their own. They are dependent on somebody else. The other point is reproduction. You see the patterns of reproduction. You see the shapes that you see in case of animals, the structural variation that you come across. This makes them quite different when you are going through the living organisms. Mostly you have main 3 domains wherein we divide our living organisms. One are those are the single cell bacteria, then you are going to have your plant, and then you are going to have your animal. So in this particular domains, animals are the ones which are highly motile, which are highly reproductive, highly complex structures, right? When you say truly animal, you mean you are talking about the animal which shows you lots of physiological aspects, all life processes starting from digestion till that of reproduction, right? You see a lot of variation and types in case of reproductive modes, lot of behavioral patterns. You will find animal eating, walking, jumping, moving, running, showing anger, having territorialism. You have lots of behaviors which animals will show, which a plant might not be able to show, which what we relate as emotions, anger, hunger, right, appetite, and also you have lots of things which, because of which all these things gather, and then you say it's an animal. The thing is that as a scientific aspect, we don't recollect that these are all the parts of science that you are studying, and that is the reason when you talk about taxonomy, it is all these points, we put it in a systematic way, and we try to study and analyze that organism in a proper scientific manner, and that's why this subject is taxonomy. So what basically taxonomy means? In Greek, taxis is organization, nomos means law, a very simple and a very initial branch of biology. And what do you do here? You do the classification of organisms, but you can see I have put one word, logical groups. So we always go in a systematic, scientific, rational way. We just don't pick up animals and put them together. We try to find out what are their characters, whether they are similar, they are dissimilar, is there any scientific rational reasoning behind it. Accordingly, we'll put them into some logical groups. And that is how we try to link up the evolutionary aspects to these animals. If you have some kind of reasoning towards evolutionary aspects, we would like to put those animals together in one particular group. And that is what is the job of taxonomy, a very old science, rather a very initial science. See, previously we never had sciences like biological science or life sciences. We started with only natural science. That only started by observations, nothing more. Darwin used to just make drawings, nothing more than that. So from drawings he tried to find out so many theories. For example, natural selection theory, right? So that is from there we started towards taxonomy, and then we haven't stopped here. Why you should study taxonomy apart from your entrance exam? There is a lot of reason behind going through this subject, and why JNU has also prescribed that you should understand taxonomy. There is also a lot of reason behind you appearing in an exam where taxonomy questions should be given. The reason is we are moving forward from taxonomy to one more science, or the modern science, or the modern aspect of taxonomy, which is what is systematics. So now generally we don't say taxonomy, we say we are studying systematics. Now why systematics? How systematics came? What actually systematics is? So you can see it's a word systema, which means systems of classification. Now systema word was there was a Greek word which was translated into Latin, and that is why it is Latinized word systema, which means these systems of classification. What is more to it which was not in taxonomy? The more parts to this particular new science is that it does not only cover classification, but it also goes in the details of identification, nomenclature of those organisms, again with a scientific background, finding out some rational reasoning, okay, clarifying more doubts which we were not doing when we started taxonomy. Taxonomy was more or less based on only the external appearances of the animal. So we were only doing morphological taxonomy in the initial time. Now we don't do that. We have went further, we go into the details of the system, we study the systems properly, and that's how you have a newer subject, that is systematics. Science does not stop here. Science still goes further, does lot of research, tries to find out more and more about the animals in detail, studying various aspects of the animals like physiology, anatomy, you have different other morphological features, reproductive patterns of the animals, and modifying these old sciences, we come across another branch of systematics which is nowadays known as new systematics. There is a lot of scopes of going further, making a career in new systematics now. Again, we don't stop here. This all story is somewhere around 1900. Now, when we have been going through this taxonomic studies, we were simultaneously into these three particular concepts also: phylogenesis, phylogeny, and phylogenetics. You must have heard about phylogenetics a lot. Nowadays, it's a very commonly utilized word in number of reference books available. But where has it come from? See, phylogenesis is the process. What kind of process? It's a process which helps you understand how an animal evolved. See, we are all trying to understand from where life has come. We still don't understand the mechanism of origin of life, and that's why a lot of subjects are trying to correlate into the evolutionary biology. So phylogenesis was one of the aspects or one of the processes which scientists could find out through the science phylogeny, that how an organism evolves, how group of organisms come into existence. When you say you see dogs, cats, buffalo, goats presently in today's era, were they present previously? How they were looking in the previous era? What was the status? When they came into existence? Why dinosaurs extinct? So all these things, when you're relating to, you're trying to understand phylogeny science, wherein you try to understand phylogenesis process, which is how the animals are evolving. Going further to it, when you study the same process at a macromolecule level, you go into details of it inside the animal's body, going deeper into the layers, try to study the proteins of it, and then try to correlate whether it had any relationship with the evolutionary aspect, then you are going to talk about phylogenetics. So it deals with the genetical aspects of your animal study, rather taxonomy. You can see how far we are moving on. Again, we don't stop here. Now, this is what we do, molecular phylogenetics, the most recent advancement towards finding out exactly where the animal stands when you are classifying the animal. And again, you are using lot of DNA sequencing. You do lot of DNA, RNA, protein, molecule studies here, and you try to find out evolutionary relationships. We also try to justify whatever Darwin had found out. Actually, those patterns of classifications are appropriate, or we need to improve. Now, this is very, very important, and that is the reason we are moving towards molecular phylogenetics. Once again, the branch wherein you can make your careers. This particular phylogenetic subject expresses itself into a phylogenetic tree. So generally you try to develop lots of phylogenetic trees related to different kingdoms, different phylums of the living organism. Now what do you mean by phylogenetic tree? This is how it looks. We are not going to do more details of this particularly when you are not into the actual taxonomic study. You have a lot of cladograms and phylograms available, but this is just for your understanding that phylogenetic tree looks like this, and this is how you try to understand how close an organism is and how separate or how different the organism is. Now here you can see I have just taken a very simple tree of life wherein you have three main domains, which is what we divide the animals or living organisms into. One is this bacteria, one is this archaea, and then you have this eukarya. Bacteria is known to you much better than me, but archaea, first of all, initially it was considered as eubacteria. Now we are moving, as in when we are studying molecular phylogenetics, we are trying to separate it because we are understanding that it's not same as a bacteria, it's something different. Even though lots of things like the cellular levels are quite similar to bacteria, but it does have its own distinct features. So we have a separate domain, which is what is archaea, and then you have your eukaryotes, wherein all the animals and plants are belonging to this particular main domain. There is nothing above the domain. When you say living organisms, you step into domain first before you start classifying them. Living organisms are always divided into these three major domains, and then you have the kingdoms coming up. which is what you know about the taxonomic stratas that you must have studied. One more branch, that is molecular systematics. It is nothing but a broader version of your molecular phylogenetics. This is the one wherein you do lot of taxonomic study along with biogeography. See, kangaroos are not found in India. They are very much into Australia. So why only in Australia? So when you are trying to find out what is there in Australia, that is the reason kangaroos are present there, or what is the reason that we have Asiatic lion and not other countries. Okay, so when you are relating your study of animal with respect to biogeography, you talk about molecular systematics, again talking at the level of molecules. We are not doing superficial studies anymore. We started from superficial studies, but this is the present status of taxonomy. It has not vanished, it has not disappeared, but it has changed into more and more better and better refined version. So molecular systematics could come only because Darwin could do those sketches. Okay, they were the ones who made the pictures of animals and birds and all, and told us what are the morphological differences. So we have travelled long from taxonomy till this of the molecular systematics. Now, why all these studies? What is the need of all these particular taxonomic studies? Why you want to undergo phylogenesis, or why you want to undergo molecular, phylogenetic aspects and all of an animal? The reason is all of these. When you study taxonomy at any level, maybe molecular, maybe superficial, at the morphological, maybe anatomical differences, whenever you are trying to understand an animal compared to animal or animal groups, you are trying to define all of these. You are trying to understand or trying to input something into all of these particular things. First of all, you classify, because it's very difficult to study all animals at a time. So that is the reason taxonomy provides a lot of conveniency in studying the animals. Whatever you want, you just want to study protozoans, okay, just be into that group. Don't switch on to mammals, right? If I give you a mixture of lot of animals, 100 animals, and I tell you that you do the study of all these 100 animals, it's not possible for you. But if I specifically tell you, you study porifera or you study a sponge, you know where you have to look into. Though it makes very, very easy for you to understand and do further research and find out more details about that animal. So convenience of study is very important. We are into science, and science is always equal to system and systematics. When you say systematics or science, it's a discipline. And discipline is always going to be stepwise, methodical, procedure-wise. And that is the reason animal study is also systematic when you are talking about taxonomy. So taxonomy provides you all the details of an animal in a systematic way. Whichever animal you are interested into, you study that, but in a very systematic way, in a scientific way. It's not haphazard. You don't have to get confused, but you have to remove your confusions when you are into taxonomy. Knowledge of affinities. What do you mean by affinities? Affinities means what? No, it's not attraction, sorry. Affinities means closeness. How close they are, how far they are. Okay? For example, human beings are very close to apes. So how do you know that what resemblance you have with apes, and what characters we are having that apes and we share? So that is what is affinity. You find certain organisms very similar, very closely related. Few organisms you find them very different. I cannot compare a small amoeba to that of a mammalian body. It's totally different. You understand that difference also. Okay, you can't compare an elephant to an amoeba. They are absolutely different and very far off. So when you are doing this taxonomic studies, you come to realize that these things are the ones which are very much close, and these are the ones which are very far off. Now that helps you to understand the sequence of evolution, which might have come first, which might have come from the common ancestor, which is what is your knowledge of phylogeny, right? This also helps you understand how they might have come through adaptation system. You have lots of different types of adaptation. Few of them you must have studied, like homologous organs and analogous organs, connecting links, These are few evidences of adaptation that you must have gone through. You also know many of the adaptations, like you are having aquatic adaptations, flight adaptations, right? Volant adaptations. These are different adaptations that an animal is going to accept, or during their evolution they are going to consider it as a part of their being, so that they evolve in that particular habitat in a proper way, and that leads to the succession of that particular species or race. So that is, these all things you come to know only and only when you study the animal in a systematic way through taxonomy. Now coming to your actual science of taxonomy, because there is always a question, it's very difficult to study, it's very boring to go through the classification. I have given you very simple 3 pages which will give you almost the whole idea of how animal kingdom has come into existence. Now what you have to do is, on the very first page of the study material given to you, you can see there is a table, animal kingdom. Can you see that? Now, if we are not going to talk about plant kingdom anymore, I have already told that there is nothing more than the domain. So, after the domain, immediately you step into one of the taxonomic strata, that is kingdoms. And we are going to concentrate only on animal kingdom, as I have chosen to be a part of zoology, right? So, if you take animal kingdom, how you will divide the whole animal kingdom? This small chart represents the whole aspect of your animal kingdom. If you can remember this much, your lots of MCQs will be automatically solved. You just have to remember these 3 pages thoroughly. If you know these 3 pages, it will help you solve your MCQs in a rational way. You just don't have to tick mark whatever comes to your mind, but you will be able to put some logic behind choosing what is correct when you are going through your MCQs. Now when you see this particular chart, you can see there are 2 words, protozoa and metazoa. Can you find that? These are the sub-kingdoms. Now this protozoa and metazoa, basically they are divided into these 2 sub-kingdoms on 2 most important characters, and that is who is unicellular and who is multicellular. Fine. So when you say protozoa, they are the ones which are single cell, so unicellular, microscopic, do not have anything like tissues. And when you are talking about metazoa, you are talking about multicellular tissue grade animals. Actually, when you say that animals are highly complex organisms, somebody told me when I asked about what are animals, they are motile, they are very complex bodies. When you say all this, you are talking about true animals. And when you are talking about true animals, you are talking about actually the animals belonging to metazoa. You are not talking about protozoans. We are still confused about protozoans. Okay, we are still into the procedures of understanding whether Euglena should be kept into protozoa, or whether it should be kept into plant science, or whether it should be a part of microbiology. Okay, so there are lots of these kind of planktonic organisms which belong to particularly protozoan subkingdom, which we are still confused with, as it's not very easy to study these organisms, and we are not able to study all these species available on our globe. Very hardly 3% of the chordate has been so far studied. Maximum studies have been done on invertebrates, and that too not at the aquatic level. If you go to the oceans, the oceanic invertebrates are pending to be studied, because we are not still clear with the terrestrial aspect. So the true animals belong to this metazoa subkingdom, and in that if you see further, it ends with infra kingdoms. There are 3 main infra kingdoms: mesozoa, parazoa and enterozoa. Right? Now if you see each of these, they have a phylum. When you see protozoa, it has only 1 phylum, and then there is no further phylogenic tree possible in it. It ends over there, right? So protozoa is very well known to you. You have already done little of the classification. You know amoeba, Euglena, paramecium belongs to this particular phylum, right? Then even in parazoa, phylum Porifera is known to you, the phylum of sponges. Okay, I would only consider those phylums which you already know, so that it becomes easy for you to understand. The rest of things you just have to know that these are also existing, so that you don't just keep off actual animal kingdom. In infra kingdom, enterozoa or eumetazoa, right? You have long classification pattern. When you say, see, Mesozoa and Parazoa, it again ends with one phylum. But when you go further into Eumetazoa or Enterozoa, it is divided into grades, which is not mentioned here. Radiata and Bilateria, these are two grades. They are known as grades. See, each taxon has a name. The name kingdom is a taxon name. Okay, when you say subkingdom, again it's a taxon name. When you are saying infra-kingdom, again it's a taxon name. Same way, you step after infra-kingdom into grades. Which are the grades here? You divide your Enterozoa into two grades, that is Radiata and Bilateria. Now these two grades are on the basis of the symmetry that an organism possesses. When you say Radiata, it's clear, it's radial symmetry. We'll discuss what is radial symmetry in the coming slides. Bilateria also means bilateral symmetry. So from those words, these two particular groups, rather grades, have been given. So all your major animals are divided into two major symmetrical groups. One is Radiata and the other one is Bilateria. Now you see in Radiata you have only two further phylums, Cnidaria and Ctenophora. Right? What is Cnidaria? Coelentrates. Now we very loosely use this word that coelentrates are Cnidaria. Number of books also write like this. Actually coelentrates are not Cnidaria. When you say Cnidaria and Ctenophora together, then it is coelentrates. And coelentrates was an old phylum term. Nowadays we don't use anything like coelentrates because we have reached to that extent to understand that this particular two phylums are very different. Which two phylums? Cnidaria and Ctenophora. Initially we used to believe that these all organisms are very similar because Ctenophora is also going to have things like jellyfish. But actually it is not so. When you go further into the details of the study, you come to realize that okay, these things are very different at a molecular level. So now we have placed these two organisms in two different groups, rather two different phylum. One is Cnidaria and one is Ctenophora. So there is nothing like coelentrates. We use very loosely, even in the references, it's very loosely utilized. But you have to remember that coelentrate makes actually two particular phylum, that is Cnidaria and Ctenophora. And nowadays you have to say only about Cnidaria. You don't have to speak the word coelentrate anymore. It's an old story to talk about coelentrates. Now when you see in bilateral symmetrical organisms, you have further classification based on the condition of coelomes. You can see here there are three patterns: acoelomates, pseudocoelomates and eucoelomates. Eucoelomates are also known as coelomates, and we very well understand these three particular terminologies. When you say acoelomates, you don't have a coelom, right? We'll also clarify further in the slides what exactly you mean by coelom. I've taken few of the concepts like that. Pseudocoelomates means you have a false coelom. The word pseudo means false. Okay, so it's not the true coelom. And when you say eucoelomates, you mean you are talking about organisms who actually possess the coelom. Now it's already well defined that when you are talking about acoelomates, you have platyhelminthes phylum. Okay, so when you are asked questions that which particular phylum has no acoelomate kind of a grade, you should be able to justify from this particular chart itself. You don't have to go in details of this. Okay, but you should remember from where the sequence starts. The coelentrates do not have anything like a coelom. So after coelentrates, you directly step into platyhelminthes flatworms. That is, I am talking about liver flukes and planaria. Okay, so you are talking about that group. From there you start about acoelomates and pseudocoelomates and all, right? Then you see esculmenthes, roundworms. What are roundworms? Pardon? Ascaris, where you will put your earthworm. Annelids, not into esculmenthes. Esculmenthes is also having a controversy. It is also the one which is commonly known as nematoda. Okay, and many a times there are a lot of classification, controversial reviews, or you can say different school of thoughts amongst the scientist, because certain scientists put organisms according to the kind of morphological feature he finds or she finds it to be very important, and accordingly there are variations in classification. So there is always a controversy about whether Aschelmantis is Nematoda or Nematoda is Aschelmantis. Actually, Nematoda is also one of the classes which belongs to this phylum of Aschelmantis. Now, you may come across a separate phylum named as Nematoda. Don't get confused, the animals are same, roundworms only, okay? But the thing is, the classification pattern might be little different compared to the pattern you might be reading in some reference because of the difference of opinion of the scientist, right? So that comes into pseudocoelomates, and then when you see eucoelomates, you see your major groups, all the rest are kept into this eucoelomates, particularly chordates. So all the chordates are going to have coelom. So there shouldn't be any confusion when you are asked a question related to coelom, existence or presence of coelom, from which phylum coeloms will start, or from which phylum you have pseudocoelom, thereafter coelom, because these are the common questions. Firstly, in which phylum coelom is being considered or is seen or is found. Then in which particular phylum pseudocoelomate or which particular phylum has pseudocoelom. These are all very common MCQs even in NET. Even when you are appearing in NET life sciences, you will have this kind of very common questions coming up, because this is the basic. If you don't know this, you can't reach to that molecular systematics. You need to know first the core of it, then only you can go inside, in the details of that particular organism. So this should be thoroughly known to you lifelong. If you know this, then only you can step into the molecular science, right? Okay, apart from this particular classification, two things are not mentioned here. See, I have already told you that classification still suffers lot of controversy. So one of the controversy is with this bilateria again. Now what is that controversy? One of the scientists divides this bilateria into further divisions. Instead of going through directly these three types of coelomates, they divided into two types of division. And those two types of divisions are protostomia and deuterostomia, which is not mentioned here, but when we will discuss further slides will come across those two particular groups. But you have to remember that there are two divisions given by some scientists in case of bilateria, okay? And those two particular divisions will divide again your animal kingdom. The remaining phylums again will be bifurcated. Now when you say deuterostomia, deuterostomia is going to have all your chordates, even echinodermates. So apart from echinodermates and chordates, the remaining one are left into protostomia, okay? So they don't divide all the time in the three different aspects, that is acoelomate, pseudocoelomate and eucoelomate. Not necessarily that you will find this pattern existing in all the reference book, okay? You might also come across one more step into bilateria, and that is divisions. Two divisions, protostomia and deuterostomia. Protostomia has all the organisms, the remaining groups till that of echinodermates, right? So what is remaining? You have from annelids till that of echinodermates. So annelids, molluscans and arthropods, this all belongs to your protostomia, right? Whereas when you are talking about deuterostomia, you are going to have phylum echinodermates and phylum chordates. That's all your vertebrates included into it, right? So if you remember this chart, you know the basics from where it starts. Now you can turn the page, come down onto the last page. There is one chart on the last page. Major characteristics of principal animal phyla. Now there is always a confusion with the students. How should we remember? There are so many systems, whether digestive system is present in cylindrate or not, whether you have in platyhelminthes asexual reproduction mode or not, what kind of symmetry they possess. So you can see here the major characters are being written in bold letters. It will start from cells, then germ layers, then organization, symmetry, segmentation, digestive tract, excretory organs, coelom, circulatory system and respiratory system. So most of the things are covered in this particular chart, and it tells you almost everything related to your invertebrate phyla. So if you can just summarize this and thoroughly understand this chart, you will never lose your marks in MCQs related to whether this particular organism has a radial symmetry or not, or whether this particular organism is a coelomate or not, whether it possesses an excretory system or not. And most of your JNU questions are coming more towards, or it's more, the flow is more towards invertebrates. So the major popular groups are covered into it. You just have to go through what is written in the small columns. Every day you just read once or twice and you will be thorough with this particular thing. We'll go through each of the concepts now, so that you understand what you mean by tissue grade and what you understand by radial symmetry. So your chart will be more clear to remember, right? So understanding the concepts and then going through the chart will help you give lot of easy logics during your solving of the MCQs in your examination. And this you have to mug up. Literally you have to mug up this if you want to score MCQs with respect to invertebrate phylums, right? So we'll come back to this once again when we clear all the concepts. We are moving further. So this is where we are presently. Now what I am doing is not discussing any classification chart, but I am taking very basic concepts which will help you understand taxonomic classification, how and when and why the organisms are being divided. So the very first is these terminologies. Those sound very simple, a lot of things to do with your embryology also. You know what is phylum Chordata, okay? It's all about possessing a notochord, right? And you also know that when your notochord in the embryonic stage, further when the organism is developing, converts into vertebral column, you are going to name those organism vertebrates, right? It sounds very simple to understand, but the basic issue is what do you mean by this notochord? So we are concentrating on what exactly notochord is, okay? So notochord, when you say notochord, it's a cord-like, a tube-like, rod-like structure which is generally found in the embryonic stages of all chordates. Now when you say chordates, you are talking about the vertebrate phylums, you are not talking about the invertebrates. So notochord is never ever going to be present in your invertebrates. It's not there in the invertebrate group. It's simply present in the vertebrate phylum. So you're talking about all your vertebrates like fish, amphibians, mammals, all of these, the reptiles, all of these, they are going to have notochord in their embryonic stages, right? What it is composed of? So it is going to be the one which is coming from cells which are derived from mesoderm and defines the primitive axis of the embryo. When you say primitive axis, see, vertebrates are the ones which have backbones. So a lot of things matter when you are having a backbone. Now when you want to have a backbone, a vertebral column, you need to have some base during your embryonic stages. Now that base is provided by these notochords, and that is the reason you cannot do dissection of vertebrate animal the way you do it with invertebrate animal. Suppose you are given an earthworm, what you do is from the dorsal side you cut the earthworm. You can do that. Just place the earthworm like this, put a cut, and you start undergoing the dissection. But when you are taking a rat, you put it upside down. You never put it straight, just as it is, and then you put a cut. The reason is you have a backbone. So that's the reason you always tilt the rat upside down, and you will put the cut from the ventral side, because you don't want the hindrance of the backbone. So backbone is a major difference between your invertebrates and vertebrates. Okay, so for the development of the backbone, notochord is must. If it's not there, you cannot have this particular backbone or vertebral column developing. Now, if in some chordates it might persist as it is, it's not necessary that all the vertebrates are going to have the notochord converted into no vertebral column. Few might not, just like amphioxus, and few might or major do the conversion of this particular notochord into vertebral column. During those time, what else it is going to play the role is, it becomes the nucleus pulposus of the intervertebral disc. This is the major role that the notochord has to play. Okay, now what this exactly is, this is how it looks. So when you are talking about vertebral column and its development, this disc, intercalated or vertebral disc, this is the one which has two layers. This outer layer is nothing but a fibrocartilage, and this inner layer is a water cum matrix kind jelly-like of a material, which is once again coming from notochord. So this is very important. You know, if there is a problem with the disc, what happens? Okay, your backbone is not going to help you out. You can't do the movement. So all the movement, the flexibility of your backbone depends on the linking of two vertebrae and the layer between these two vertebrae formed by this particular intervertebral disc, which is actually once again a part of notochord. If you ask me to go in details of this, I'm sorry, I'm not still further going, because for that you need to understand embryology. Now it's very difficult to bring all those topics here in this short period of time, but it's very interesting if you can go back to your embryology and find out more about the notochord, right? So through notogenesis, process happening during the period of embryology or embryogenesis, you have one phase wherein notochord is developed. So that development of notochord is known as notogenesis. And when does this particular phase happen? It happens during gastrulation. Okay, we'll come to gastrulation also, what exactly gastrulation means. But this is where your whole system, when you divide chordates and non-chordates and invertebrates and vertebrates, notochord plays a vital role into it. So you should understand where exactly this notochord is coming from. This is how it looks in the original way. You can see here, this is a dissected small tiny lungfish, and this portion over here, this is what is your notochord present, right? Now those were chordates and vertebrates. What are non-chordate and invertebrate? Animals lacking a notochord are all known as non-chordates, but animals lacking a vertebral column are known as invertebrates. And many of the books, including students, they use these two terms very loosely. Non-chordates and invertebrates are same. It's not so. Okay, invertebrates can be into non-chordates, but non-chordates are not necessarily going to be into invertebrates. You have a major difference. The difference is, if you have a lack of notochord, then only you can say it's non-chordate. Technically it is like this. Even though we might be using loosely interchangeable terms, many a times we loosely say these are all non-chordates, but then you have to actually see technically whether they are really non-chordates or not. Same way invertebrates. When you say invertebrates, you are talking about only vertebral column, right? So this is the two major thing which you must understand when you are doing classification of animals. Now there are certain invertebrates which might not have a development of vertebral column, but they can be put into chordates because they show notochord presence. Or rather, I would say there are organisms which have presence of notochord in their life, throughout their life, but they will never convert their notochord into a vertebral column. Now those kind of organisms, we put it into protochordates. So you have protochordates as a special group available. In that you have these three major groups: hemichordates, urochordates and cephalochordates, which have the notochord persistent throughout their life, right? There are a lot of other differences, but then they have persistently their notochord present throughout their life, never converting it into the vertebral column. So you need to know non-chordates, chordates, protochordates and invertebrates and vertebrates. These 5 terms should be very much clear to you. Then only you can go further into the science of animal taxonomy. Now to help you out that, I have given you one comparative table which talks about invertebrates, vertebrates in general. This also you must know it thoroughly, so that it helps you in the MCQs, because all this will help you have a judgment related to the options given to you. It's very important how do you judge according to the concept clarity. Then only you will be able to tick mark the actual answer, otherwise you will get confused. So for that you need to go through these particular tables. Even if you do these many 3 pages, I am sure you will be able to score very well into taxonomic study questions if they come up into your MCQs. Now it's your test. You tell me whether when you minus invertebrates, from invertebrates protochordates, what exactly you are going to get. What should be the answer? invertebrates minus protochordates, then what kind of group you will get? Pardon? Non-chordates, right? Okay. Another question. All the non-chordates are dash, whereas all the invertebrates are not dash. Think of it. The answer lies in your question only. All the non-chordates are? Very good. All the non-chordates are invertebrates, whereas all the invertebrates are not non-chordates. So this confusion you have to clarify and remain clear. Don't get confused out of this. Moving further to the next concept, that is symmetry, a very important aspect of basic taxonomy, and it is the one which deals with arrangement of body parts. Some kind of symmetry we try to find out with respect to geometrical shapes, with respect to geometrical designs. Basically what we do is we refer to the division of the body into equal parts by some lines or some planes, whether we can do that or not, and accordingly we have defined few of the symmetries. Now when you can divide an animal into equal parts, at that time the animals will be known as symmetrical. But when you don't do that, or when you cannot, just like an amoeba or a sponge, if you take one sponge, if you take one amoeba, you cannot just put them into appropriate planes, then they are going to be known as asymmetrical. So animals can only be divided into symmetrical and asymmetrical groups. Now there is nothing more to asymmetrical, but there is lot when you talk about symmetrical groups. So basically we have this three types of symmetrical groups: spherical symmetry, radial symmetry, and bilateral symmetry. Now bilateral symmetry, you must have drawn lots of freehands. You know, when you do it in the drawing, one side you draw and then you put a paper and then you are going to draw it, press it on the other side, because you want the exact mirror image of it, right? So bilateral symmetry is something related to it. So you have one plane going through it and you are going to have your bilateral symmetry. But when you see radial and spherical, there are a lot of changes. So what are the changes? You can see that in generally spherical symmetry, you are going to have center, one point, because spherical symmetry is possessed by the organisms like volvox. Okay, now these organisms, they are going to have a center from which lots of planes can be divided, right? So that's why this is spherical symmetry. It's very much restricted to lower organisms, particularly protozoans. In protozoans and the classes of protozoans, you will find organisms having this kind of symmetry, but not in the further groups. Most of the groups, they are going to be with respect to radial and bilateral symmetry and all. This is how radial symmetry grows from a central axis. Now here you have a central axis, here you have a center point. So that's how they are different. Okay, passing from a point, passing from an axis makes things different. So here you have a central axis and it's just like cutting a cake. You divide it into lots of groups and you have equal pieces available. Something similar to that, you're going to have it with radial symmetry, and the photographs that you see here, all these organisms are going to show you radial symmetry. So you have the starfishes and sand dollars. So lots of your echinodermids, they are the ones which are going to show you radial symmetry. You're also going to have lots of coelentrates like sea anemone and all, showing you radial symmetry. There is one more variant of this radial symmetry which is known as biradial symmetry. So that's also another particular variant which is bilateral symmetry. Now here the only limitation is, the most important feature is, you can divide your organism only in two planes. One going through longitudinal and sagittal axis and one going through transverse axis. You can't have more planes here, which is not the condition when you talk about radial symmetry. So again you have a difference when you are talking into radial symmetry. Now radial and biradial are also very loosely placed into the books. Many a times the same photograph, when you go on the internet, you will find the same photograph representing biradial symmetry. So I have to tell you that we are still not clear whether this should be going into a radial or it should be going into a biradial, right? So you have to be very, very smart when you have question wherein only radial is given, and you have a question wherein radial and biradial is given. So when you are given biradial, you have organisms like sea anemones. You have to be specific whether they have mentioned about sea anemones or they have just mentioned about coelentrates or Cnidaria. If it is so, and if there is no option of biradial symmetry, you can select radial. But if you have an option of biradial, you must go for biradial because that suits more to the sea anemones. So you have to be very smart in selecting your answer according to the presence. Now when you see here, you have two particular groups, Ctenophora and Anthozoa. Now Ctenophora you already know, it's the one which we separated from the Cnidaria. It's a very separate phylum. But what is Anthozoa? It's one of the classes of your, once again, the coelentrate, basic coelentrate group. So ultimately everything is belonging to that Cnidaria kinds. So some part of Cnidaria, those organisms, and all of your Ctenophora, these organisms are biradially symmetrical. So when you say Anthozoa, which organisms? Most of the sea anemones, they are the ones which belong to Anthozoa and they are the ones to show you biradial symmetry. So if you are talking about the organisms other than this, or an example comes in your exam other than this, you should be clear enough to find out which is the answer. Okay, in nowhere it's going to be bilateral, nowhere it's going to be spherical. But you should be very much smart enough to select whether it's biradial or it's only radial. So that's where this particular thing helps. Now what kind of organism is this Ctenophora? So just for your understanding, I am showing you this one of the ctenophoran organism. It looks like this. So it's very different from what you see in Cnidaria. Bilateral symmetry, the most common one which we understand, all your higher animals are going to show you this particular bilateral symmetry, and you can see that you have this sagittal plane as well as you are going to have a transverse plane possible over here. So that's why this bilateral symmetry is there, which particularly longitudinal axis. There you had central axis when you talked about radial. When you talked about spherical, you had a center point. Now you are talking about longitudinal axis from which you are going to divide your organism. So that is the reason it is bilateral. You have two sides, equal sides available over here, right? So bilateral symmetry. Now it's your turn. You tell me this beetle has what kind of symmetry? Bilateral. What about this coral polyp? Radial. And sponge? Asymmetrical. It doesn't have any kind of symmetrical organization. Very important third concept, that is coelom. Now we very many times write body cavity coelom, but again there is a difference technically. When you say body cavity and coelom, you are talking about two different things. Body cavity only means internal space present inside the body. It doesn't mean it's always coelom. So you have to be very, when you read your books, you check what exactly they want to say, because many a times they might be telling you about coelom and writing body cavity, or they might be talking about body cavity and using the word coelom. So you shouldn't get confused about it. Coelom is actually the true body cavity, which is a fluid filled space. Where does it exist? It lies between outer body wall and inner digestive tube. So you have a digestive tract, you are having the actual body wall, and the internal space between these two is what is your coelom. It arises as a secondary cavity between two layers of your embryonic mesoderm and contains most of the visceral organs. What do you mean by visceral organs? What are visceral organs? All your main organs, okay? Heart, lungs, stomach, pancreas, intestine, these are all your main organs. So all your visceral organs are the main organs of all of the main systems. This is our concept of acoelomate, pseudocoelomate and coelomate. Apart from this particular entrance exam, because you have very less time, maybe for this exam it's not possible for you to undergo all the embryological studies, but it's my humble request, if you want to do something with respect to molecular phylogenetics or with stem cell studies, better read embryology. It's very important. All your concepts lie into embryology ultimately. From where does it arise? So if you are doing any kind of taxonomic study in your life, at any moment or at any point of time, please study embryology thoroughly, because that's the way you will be able to find out your answers properly. Now when you see look back into embryology, you will find these three particular layers which you name it as germ layers. So ectoderm, mesoderm and endoderm.